This is part 2 of the Mineral Nutrients Lecture 4, which is part of the plant physiology subject, which is offered in the NMIT Agriculture and Viticultural degrees. My name is Nikki Cooley. Today, in this part of the mineral nutrients, we're going to deal with nutrients and soil and nutrient uptake by plants. Please ensure that you have watched part 1 before you watch part 2. Mineral nutrient elements differ in 3 ways: their functions, their mobility, and their characteristics, deficiency or toxicity symptoms. As an overview, it is worth noting that crop plants typically use less than half the fertilizers applied to soil. There are problems such as leaching into soil waters or groundwater. The nutrients can attach to soil particles, and the attachment can be stronger than the pull by the plant, and they can also contribute to air pollution, atmospheric nitrogen deposition. So let's begin by understanding how clay particles attract ions. A clay particle is covered with negative charges, and these negative charges are called anions. The image on the screen shows you a theoretical soil clay particle, and these are the negative charges that surround it. Elements with positive charges, which many nutrients have, will be attracted to the compounds with the negative charges. Therefore, elements tend to adhere to the clay particles due to this attraction. Root hairs secrete hydrogen ions into the water around the nearby clay. So this is all the solution around the clay. The smaller hydrogen ions, cations, can replace the larger macro and micronutrient cations. Therefore, the nutrient is released to and available to the plant root for uptake. We deal more with this on the lecture of solute transport. I would like you to please stop this lecture and now watch the following video with the link on the screen to understand nutrient interactions from clay particles to root hairs in more detail. These slides show 2 visual representations of nutrient and element uptake of essential compounds in plants. I like both of these because they sum up the important components. Here in the soil, around the clay particles, are the nutrients. Here are the root hairs, of which they are absorbed through. Once they are absorbed into the plant, they are translocated into the leaves, where light and energy allows them to conduct photosynthesis. The energy from photosynthesis enables them to put storage compounds or to continue producing fruit. Here is a summary of plant root systems. There is an ability of plants to obtain water and mineral nutrients related to the capacity to develop the root system. In 1930, Demeter estimated from a young single rye plant 30 million primary and lateral root axes. These were 500 kilometers in length, 200 meters squared of surface area. Also 1,100 root hairs, providing another 300 meters squared of surface area. This shows you just how impressive a root system can be, and in fact needs to be. Desert mesquite roots may grow downwards more than 50 meters. Annual rock crop plants can grow between 0.1 and 2 meters in depth. If well fertilized and watered, plant root growth becomes only carbohydrate limited, and a small root system supplies for the plant. This is important from an agricultural perspective. The shift in allocation from roots to shoots often results in higher yields. It is important for us to understand the impact of root structure on nutrient uptake. Let us look at monocots. There are 3 to 6 primary root axes from the germinating seed. Plants that extend new adventitious roots are called nodule roots. Eventually forms a fibrous root system. All roots have the same diameter. In the illustration in figure 5.7, you can see an illustration of roots that have developed in dry soil and roots that have developed in irrigated soil. Irrigation management is very important in understanding how you can alter root growth patterns and therefore nutrient uptake. Dicots develop root systems with a main single root axis, a taproot which may undergo secondary thickening. Lateral roots develop from this main axis and they develop into branch root systems. Here we have an illustration of two dicots, sugar beet and alfalfa. On the slide we can see more detail about root development and root structure. The development of the root system depends on the activity of the root apical meristem, this region here, and the production of lateral root meristems. You will see that there are differences between dicots and monocots. Let's talk a little bit about the meristemic zone. This zone is where cells divide in both directions, towards the base to form cells that differentiate into tissues of the root, and towards the apex of the root cap. The root cap protects the meristemetic cells and secretes mycelium which surrounds the root tip. It may provide lubrication, it protects the root from desiccation, it promotes the transfer of nutrients to the root, and affects interactions between roots and microbes. The quiescent centre is a centre region of the meristem where cells divide slowly or not at all. The elongation zone is typically 0.7 to 1.5 millimeters from the root apex. Cells elongate rapidly and produce a central ring of cells called the endodermis. The endodermal wall becomes thickened and suberin deposited on the radical walls to form the Casparian strip. The Casparian strip is a hydroponic structure that prevents the apoplastic movement of water across the root. These processes are again explored more in solute transport lecture. Exploring root development further, the endodermis divides the root into two regions, the cortex towards the outside and the stele towards the inside containing the phloem and the xylem. The maturation zone is where root hairs appear in this zone. Large surface area for absorption of water and solutes, and they are also involved in the anchoring of the root into the soil. It is in the maturation zone where the xylem develops the capacity to translocate water and solutes to the shoot. Let us talk about the absorption of mineral ions. The precise point of entry of many minerals into the root is still under considerable debate. Calcium absorption in barley is considered in the apical region. So is iron absorption in barley. Whereas in corn, calcium and iron absorption is believed to occur over the entire root surface. Potassium nitrate, ammonium and phosphate freely absorbed by entire root surface. Ammonium in corn and rice are absorbed in the root apex. Root hairs most active in phosphate absorption in several species. Nutrients move to the root surface by bulk flow and diffusion. Bulk flow is carried by water moving through the soil. The rate of bulk flow depends on the rate of water flow, which depends on transpirational rates and nutrient levels in the soil. Mineral nutrients move from high to low concentrations, and this is described as diffusion. Nutrient uptake by plant lowers concentrations at the root surface, generating concentration gradients in the soil. When diffusion is too slow, a nutrient depletion zone forms adjacent to the root surface. Approximately 0.2 to 2 millimeters, depending on nutrient mobility. Nutrient availability influences root growth. Root growth is minimal in poor soils because the roots become nutrient limited. As soil nutrients increase, roots proliferate. Where soil nutrients exceed the optimal level, root growth becomes carbohydrate limited and eventually ceases. To give you some example, 3.5% of the roots in wheat and about 12% of the roots in lettuce are required to supply all the nutrients. Thus, the plant diminishes the allocation of its resources to its roots and increases allocation to the shoots and flowers. This resource shifting is one mechanism by which fertilization stimulates crops' yields, and these are very important concepts that are required to be understood by all people working in agriculture. This brings us to the end of part 2, mineral nutrients.